Good day, everyone. My name is Bar Michel, a year 3 student of Biomedical Laboratory Sciences at Ines Ruhengeri University. My presentation will be on the principles of vaccination and the preparation of vaccines. This is an important topic in immunology that highlights how we use the immune system to fight diseases. Don't forget to subscribe to my YouTube channel and like this video to support more educational content. Let's dive right in. What makes a vaccine effective? For a vaccine to be effective, it must have the ability to, number one, trigger an immune response. A vaccine works by teaching your body to recognize harmful germs, like viruses or bacteria, without causing illness. This training prepares your immune system to fight the real germ in the future. Number two, the vaccine must be able to mimic the germ. Vaccines use safe versions of germs, like weakened, dead, or part of the germ, to help your immune system practice without risk. The vaccine should have, number three, should have a long-lasting protection. A good vaccine provides immunity that lasts for months or years, ensuring your body is prepared when needed. An effective vaccine must be safe for everyone. Number four, it must be safe for everyone. It must be safe for most people, causing only mild side effects like sores, arm, or mild fever, while protecting you from severe diseases. An effective vaccine must prevent serious illnesses. An effective vaccine reduces the chances of severe symptoms or complications if you get exposed to the actual disease. An effective vaccine must, number six, reduce spread. When enough people are vaccinated, it creates herd immunity, which protects everyone, which protects even those who can't get the vaccine, like newborns or people with weak immune system. An effective vaccine must work for many people. A vaccine should be effective across different ages, groups, genders, and ethnicities to benefit the entire population. An effective vaccine must be, number eight, easy to use. An ideal vaccine should be simple to store, transport, and administer, making it accessible worldwide, even in the remote areas. An effective vaccine must have the ability to tailor to the disease. It targets the specific characteristics of the disease, ensuring it matches the way the germ spreads and behaves in the body. Lastly, an effective vaccine must be trusted by communities. For a vaccine to be effective, people need to trust it and be willing to get vaccinated. Public education and transparent research build this trust. So we have been talking about what makes a vaccine effective. Now we'll be talking about how do mRNA vaccines work. mRNA vaccines use a synthetic messenger RNA molecule to instruct cells to produce a specific antigen which triggers an immune response. There are a few steps involved. The steps involved are, number one, mRNA delivery. The mRNA is encapsulated in lipid nanoparticles for protection and efficient delivery to cells. Number two, translation. Once inside the cell, the mRNA is translated by ribosomes into the target antigen, for example, like viral protein and spike protein. Number three, antigen presentation. The antigen is processed and presented on the cell surface via MHC molecules, activating T cells. Some of the antigen is also secreted, triggering B cell activation and antibody production. Number four, immune response. Cytotoxic T cells recognize and destroy infected cells, while helper T cells assist B cells in producing antibodies. Number five, which is the last step, memory formation. The immune system retains memory B and T cells, enabling a faster and stronger response upon feature exposure to the pathogen. mRNA vaccines are highly adaptable, allowing rapid development for new pathogens. Now that you know how mRNA vaccine works, at this point, we will be looking at why are adjuvants used in vaccines. Firstly, what are adjuvants? Adjuvants are substances added to vaccines to enhance the immune response to the antigen. They are especially important in vaccines containing weaker or subunit antigens. What are the functions of adjuvants? Number 1, it boosts immune activation, stimulates innate immunity to enhance antigen presentation and adaptive responses. Number 2, it prolongs antigen exposure. It creates a depot effect, ensuring a sustained release of the antigen. Number 3, it enhances antibody production. It increases the quantity and quality of antibodies. Number 4, it promotes T-cell responses. It encourages helper T-cell differentiation and cytotoxic T-cell activation. Number 5, for dose sparing. It reduces the amount of antigen needed per dose, allowing more doses to be produced. Examples of adjuvants. Number 1, aluminum salt. Aluminum salt is commonly used in inactivated and subunit vaccines. Number 2, oil in water emulsions. Oil in water emulsions is used in influenza vaccines. Number 3, toll-like receptor agonist. Toll-like receptor agonist is a mimic pathogen-associated signal to stimulate innate immunity. Adjuvants are critical for vaccine efficacy, especially for antigens that are weakly immunogenic. In the previous chapter, we have been discussing the principles of vaccination. Now we will be diving into the preparation of vaccines. How are live attenuated vaccines prepared? How are live attenuated vaccines prepared? Live attenuated vaccines are made by weakening a pathogen so it can still replicate but cannot cause disease in healthy individuals. The process involves, number 1, serial passage in culture. The pathogen is grown repeatedly in non-human cells or suboptimal conditions, causing it to adapt to these environments and lose its ability to infect human effectively. Example, the measles, mumps, and rubella vaccines. Number 2, genetic modification. Specific genes responsible for virulence are deleted or inactivated, rendering the pathogen less harmful. Example, rotavirus vaccines. Number 3, which is the last, temperature adaptation. The pathogen is adapted to grow at lower temperature, making it unable to thrive at human body temperature. The resulting vaccine retains immunogenicity, inducing a strong and long-lasting immunity. Stay on the preparation of vaccines. We will be looking at the advantages of subunit vaccines. Subunit vaccines contain only specific antigen like protein and polysaccharide from a pathogen rather than the whole organism. The advantages of subunit vaccines are, number 1, safety. No live pathogen, so there is no risk of causing the disease. Number 2, targeted response. It focuses the immune system on specific antigens, reducing off-target effects. Number 3, it has fewer side effects. It lowers the likelihood of adverse reactions compared to live vaccines. Number 4, stability. It is stable. It is often most stable during storage and transport than live vaccines. Number 5, compatibility with immunocompromised patients. It is safer for individuals with weakened immune systems. Number 6, which is the last, is customizability. It is easy to include multiple antigens or tailor to new strains. Example, multivalent HPV vaccines. Thank you for watching, guys, and I hope this presentation has helped you appreciate the science behind vaccines and their importance. Don't forget to subscribe to my YouTube channel and like this video to support more educational content. Thank you for your attention and feel free to ask any question. Bye.